So we are recording now. Good afternoon, everyone, and thank you for joining us. I am Amy Harrell, the ACRM Neuroplasticity Networking Group Secretary. For the sake of sound quality, I'm going to be muting everyone's line during the presentation. And guests, please keep your devices on mute during the talk, and then also keep audio to one device, either your phone or your speaker. It's my pleasure to introduce Dr. Yufen Chen to you today. I've had the pleasure of collaborating with her on concussion neuroimaging projects taking place at Northwestern over the past few years, which she is going to touch on today. She's an amazing colleague, and I'm delighted for her to share her work with you. So just allow me to give a few words about her background. Dr. Chen received her PhD in biomedical engineering from Northwestern in 2008. From there, she completed a postdoctoral fellowship at the University of Pennsylvania. She returned to Northwestern in 2011, where she is currently a research assistant professor in the Department of Radiology. Dr. Chen parlays her extensive experience in magnetic resonance imaging and in arterial spin labeling in particular to many areas of study at Northwestern, including concussion. So today, Dr. Chen will be taking questions at her talk. You can write your questions using the chat feature to go in the GoToMeeting, and I'll moderate them. And then I'll unmute your line so you can expand on your questions during that time. So with that, I'll let Dr. Chen present her work.
Great. Thank you for the kind introduction, Amy. Hello, everyone. I'm Jennie Chen, and I will be talking about vascular health after mild traumatic brain injury today. So here's a brief outline. I'm going to start out by discussing measures of vascular health, including resting perfusion and cerebral vascular reactivity. And then I'll talk about a few recent publications that applied these measures to study mild TBI. And finally, I'll discuss some of the exciting results from our female athlete study. So resting perfusion refers to the delivery of oxygen and nutrients to tissue via capillaries. The average diameter of the capillary is about 6 to 8 microns. So with MR neuroimaging techniques, we can't visualize the individual capillaries, but we're really looking at a distribution of them in the brain. Perfusion is typically reported in units of milliliters per 100 gram of tissue per minute. And there's immense interest in perfusion because we know perfusion disruption leads to irreversible tissue damage within minutes. It's a major contributor to the gold signal, and it's also altered in many diseases. So it's a very important metric for brain health. So because of our continued interest in perfusion over the years, many different modalities and techniques have been developed to measure perfusion. I'm just going to focus on the non-invasive MRI technique called arterial spin labeling today. So in the typical ASL experiment, there's always a tag measurement where you invert the blood spins flowing into the brain. So when they arrive at the brain, they will decrease the total magnetization by a small amount. And then you can do a control measurement where the blood spins are not inverted. So when they flow into the brain, they actually add to the total magnetization. And when you do a subtraction between these two conditions, you end up with just a blood signal because the background tissue signal subtracts out. Now, this delta M is actually very small. It's only about 1% of the raw background signal. So when you're doing an ASL experiment and you have that urge to, play around with your parameters, increase resolution or increase scan time, this is something you have to keep in mind. The biggest advantage of ASL is that it uses your own blood spins as a contrast agent. So there's no injection required. It's entirely non-invasive. But it's important to remember that this manipulation of blood spins is only temporary. So anytime you change the magnetization of something in a strong magnetic field like an MRI, it's going to try to return to its equilibrium condition. And the speed at which it does that is governed by the T1 of the spin species that you're looking at. So in our case, this speed is governed by the blood T1, which is about 1.6 seconds at 3T. Basically, this means you lose about 60% of your ASL signal in 1.6 seconds at 3T. So fast imaging is critical for ASL. But if you're a glass half full kind of person, you'll see that the flip side of this is that the signal replenishes very quickly. So if you need to run multiple ASL scans within a single session, say, for example, you're interested in the effect of a drug on blood flow or a stress test on blood flow, you can easily do so without having to worry about things like contrast clearance. So I mentioned the ASL signal was very low, and you can see that here. These are 30 consecutively acquired subtraction images between control and tag pairs. And you can see there's a lot of fluctuations. It's very noisy. But if you average it over time, you get a map that looks like perfusion, and it has excellent correspondence to what we see in histology data, where there's high distribution of a dense distribution of capillaries in gray matter versus white matter. And you can easily convert this map into physiological units by just scaling it with some additional measurements. So there are many ASL variants out there. I'm just going to focus on the three main classes. The first one is continuous ASL, and this begins with a very long inversion RF. So tau here is on the order of 2 to 3 seconds. This is applied to a thin plane located below the brain. So any blood spins that flow through this plane during tau will get inverted. And then after that, you just have to give it enough time to flow into the brain. So there's usually a delay called post-labeling delay after the period of tau. Sometimes you'll hear people talk about CASL as a time-based tagging technique because anything that flows through the plane during this inversion RF period will be inverted. And then there's pulsed ASL techniques where you just apply a short inversion pulse to a large slab of tissue located below the brain. So everything in this slab will get inverted, but of course only the blood spins will then travel into the brain and affect your ASL measurements. So in pulsed ASL, the inversion time here is only on the order about 15 milliseconds. But your bolus of inverted blood spins is governed by the time it takes for blood spin to flow from the bottom edge of the inversion region to the top edge of the inversion region. So denoted by capital T in this schematic. And then after inversion, it's the same deal. You have to give it enough time to arrive in the brain. So in post-ASL literature, usually we use TI to denote this time, but in theory, it's the same as the post-labeling delay in continuous ASL techniques. Now, continuous ASL techniques typically have higher signal-to-noise ratio because you are inverting more blood spin, so you end up with more ASL signal. But the problem is with modern MRI systems, due to safety concerns, they typically do not allow you to run a continuous RF field for seconds long. So in order to get around that restriction, researchers have come up with a pseudo-continuous ASL technique where the continuous RF is broken up into thousands of little 500 microsecond long pulses, and you just have to make sure you adjust the phases for these pulses to maintain the inversion. But otherwise, it's a similar mechanism as traditional continuous ASL techniques. So today, PCASL was really the state-of-the-art recommended technique used for ASL because it retains the high signal of continuous techniques, but is also able to be used in today's modern MRI systems. And it also has better sensitivity and stability compared to pulsed ASL methods. And I can't stress enough that the post-labeling delay or the inversion time is very important for your ASL experiment. So here's a graph that shows you why. The red line here is the blood signal in the control experiment, and the tag experiment is shown in blue. The separation between these two lines is your ASL signal. So obviously the maximum signal difference between these two conditions is immediately after the inversion. But remember, blood takes time to travel through, your system of blood vessels in order to arrive in the brain. So you'll need to wait a sufficient amount of time to do your acquisition. But keep in mind that the signal is continuously decreasing as you wait longer and longer. So here are some maps on top that were acquired at different post-labeling delays. So you can see at very short delays, all of the signal is really within large blood vessels, and this is not really true perfusion. But as you wait longer and longer, you'll start to see gray matter filling out with signal, and very late in time where you start to see signal and white matter. So this is the reason why most ASL literature do not talk about white matter perfusion, because blood arrives so late that by the time it gets there, there's really not enough signal left for us to get an accurate measure of blood flow. Now, for normal people, we have a pretty good idea of the optimal post-labeling delay. But when you're scanning a patient with cerebral vascular disease or even collaterals, it's hard to say. And you may have to run several ASL scans at different post-labeling delays to determine the optimal parameter for your particular patient. The biggest problem in ASL is when you see an area of compromised flow, you can't distinguish whether it's actually compromised flow or it's because of delayed blood arrival, which is why currently in the field there is a big push towards acquiring multi-inversion time or multi-post-labeling delay data, which will allow us to map out both transit delay and perfusion to get rid of this confound. So in summary, ASL is an entirely non-invasive technique, and it offers perfusion in quantitative units. It does take a while to acquire an ASL scan because the signal-to-noise ratio is low. Usually you need at least 3 minutes to get a decent blood flow map. But the ASL preparation module is very flexible in that it can be combined with any type of acquisition as long as it's fast enough. And the current white paper in ASL actually suggests using a 3D acquisition so you can make use of background suppression. And this is important because remember, the ASL signal is only 1% of the raw signal. So if there is any sort of fluctuations in the background signal, it's most likely going to be greater than your ASL signal. So to be able to remove that background signal early on is useful for an ASL experiment. And then finally, transit delay is a huge confound for ASL data, which is why it's recommended to acquire post-labeling delay data to map out both perfusion and transit delay. So moving on to cerebro-vascular reactivity. This refers to the response of blood vessels to a vasoactive stimulus, and it has been shown to occur with a similar mechanism to fold response to cognitive function. So it could potentially give you supplementary information about brain function. Studies have shown that CBR is reduced in cognitive impairment and that it actually correlates with scores such as the MMSE, and it can also predict the conversion from MCI to AD. So it's a pretty important metric to look at brain function. Now, there are different types of vasoactive stimulus that you could use. The first one is to reduce systemic pressure, although this is actually very rarely done in reality because it's potentially dangerous for patients with compromised flow to begin with. In clinical settings, they tend to prefer using acetazolamide, which is a vasodilator. An injection of acetazolamide will induce increased perfusion throughout the gray matter regions of the brain. The problem with acetazolamide is response in subjects is very variable, and there are significant side effects, some of which are very long lasting. For research settings, we tend to prefer to use carbon dioxide because it's non-invasive and it's well tolerated by subjects with very few side effects, including dizziness or shortness of breath, and these side effects typically resolve very quickly. But most importantly, carbon dioxide reliably alters blood flow, so which is why researchers tend to prefer using CO2. And there are several different methods you could use to induce hypercapnia. The easiest one is to just ask your subjects to hold their breath. You don't need any additional equipment, but obviously this requires subject compliance, and your subjects need to be able to hold their breath for the entire duration of that period. You can also make them breathe CO2-enriched gas. This does require additional equipment, and you can see an example of the setup on the right in this picture. The bags contain 5% CO2 mixed with medical air, and these are connected through tubing to a mouthpiece that is given to the subject. They also have a nose clip that makes sure they only breathe through the mouthpiece. And then at the certain times during the experiment, the researcher can open or close the valve on the top of this bag to deliver the CO2-enriched gas versus medical air. And then more recently, they've developed more sophisticated setups where you can use a computer-controlled system to fine-tune end-tidal CO2 targeting, because in the second experiment where you're delivering enriched CO2, depending on the subject, because this can induce different levels of end-tidal CO2. But if you use a computer-controlled model, then this will give you a more precise control. But of course, this is a more complicated setup and definitely a lot more expensive. So once you deliver your vasoactive stimulus, you will then have to decide how to measure the response. The traditional technique is transcranial Doppler, or TCD, and this technique measures changes in blood velocity. So it gives you a global view of how blood flow to the brain responds to the stimulus, but it doesn't give you any spatial information. You can, of course, use any sort of neuroimaging techniques such as PET or SPECT and MRI. With the PET techniques, of course, there's the concern about ionizing radiation, and also these tend to have poor spatial resolution. MRI can give you both blood velocity measurements using phase contrast or imaging techniques such as ASL or BOLD. So ASL to measure CDR, of course, is a no-brainer because this is a blood flow response, and ASL measures blood flow. But keep in mind, the data is going to be noisy because of the low SNR of ASL techniques. And also remember, you need to acquire 2 images to get 1 perfusion image. So ASL also has lower temporal resolution compared to bold imaging. And this is why most CDR reports that you see in the literature use bold imaging for their measurements. So focusing in on the MR imaging-based techniques for measuring CVR, namely bold and CVF. So you can see example CVR maps acquired using bold on top and CVF in the bottom on the right. These are for low, medium, and high levels of CO2. So you can see the increase in CVR as you increase CO2, and also that the bold response is cleaner than the CVF response. And in terms of the experimental setup, CVR experiments are just like any of your task-based bold experiments. You need to have a control condition and a hypercapnia condition so that you can contrast between the 2 conditions. So if you're delivering CO2 or using end-tidal CO2 targeting, you can use these longer boxcar conditions. But if you're doing a breath hold, then of course these hypercapnia blocks need to be shorter so that your subjects can actually hold their breath during the entire time. And you repeat these a lot more times to do signal averaging. And then once you acquire your CVR map, you can actually normalize it by the change in end-tidal CO2 that will take away some of the between subject variations and improve your sensitivity. So here are some time courses acquired with CVF and bold for different levels of CO2 challenge. You can see the CVF one is definitely noisier, but notice the range of signal change is also very different. So for the blood flow signal change, it's a lot bigger than the bold signal change. So now that we kind of understand how to measure blood flow and CVR, let's see how these techniques have been applied in the field of concussion research. So first of all, why do we want to look at blood flow and CVR and concussion? Well, we know CVF is very carefully balanced in the brain and sensitive to a lot of different parameters. Studies have shown using various modalities that both CVF and metabolism change after severe brain injury, but very few studies have been published in the field of mild traumatic brain injury. And then also studies in normal aging shows us that even though your blood flow may seem normal at rest, it doesn't mean your vasculature is healthy enough to support the added demands of functional activity. So in elderly subjects, because their vasculature, their vessels are stiffer, they can't expand as readily and as much as younger people's vessels. And so they'll usually see a smaller bold response in functional activity and sometimes even have compromised cognition. So I did a quick search of CVF and concussion on PubMed, and it looks like there's a general upward trend in the number of publications since the 1980s. But if you zoom in to arterial spin labeling applied in concussion, the number of hits were a lot lower. I only found 9 papers. And if I looked for CVR and concussion, it's even less. So obviously I don't have time to go through all of the papers, but I just wanted to present a few of these publications just to give you an idea of where we stand currently in applying these techniques in concussion research. So this first paper by Stevenson Dahl was published earlier this year looking at blood flow changes in sports-related concussion at adolescents who were scanned within 2 weeks and after 6 weeks post-injury. And they were compared to 15 never concussed controls. The researchers in this group collected their ASL data using pseudo-continuous ASL and normalized each subject's CBF map by their whole brain blood flow value. And I think this is a very important step because there's a lot of between subject and also within subject variations in whole brain blood flow. And to be able to have the sensitivity to be able to detect differences between groups, it would be wise to remove some of these variations by normalizing your data. So the authors reported that there was significantly increased blood flow in the concussed subjects compared to the controls in 2 regions of the brain, the left insula and the left dorsal anterior cingulate. So for the left insula, only visit 1's data was significantly higher than the controls. Whereas for the left dorsal anterior cingulate, both visits had increased, both visits for the concussed subjects had increased blood flow compared to the controls. And then they also segregated their visit 2 data into those with persistent symptoms versus those without symptoms and compare those subjects to the controls. And they found that those with symptoms had elevated blood flow compared to those without symptoms, which are still higher than the controls. Okay. In this next paper by Barlow et al. A year earlier, this was in a younger cohort of 8 to 18-year-olds. They recruited 51 concussed subjects and compared it to 21 controls. The images were acquired at 40 days post-injury, so this is at a much later date after they sustained the injury. They found significant differences in global CBF. The symptomatic subjects had the highest global CBF, followed by the controls, and then the asymptomatic ones had the lowest global CBF. And then the scatter plot on the right is showing the correlation between neurocognitive index versus cerebral blood flow for the controls in blue versus the concussed subjects in green. And there was a significant negative correlation between these 2 measures only for the concussed subjects, not for the controls. And finally, this image is just showing the specific areas that had increased perfusion in the symptomatic subjects compared to the controls on the left versus areas that had decreased blood flow in the asymptomatic subjects compared to controls. So you can see immediately, just with the 2 papers, we already have a little bit of discrepancy between the two studies. Whether this is a function of the different time points at which they collected their data or the different age groups that they're looking at requires further investigation. And then this third paper by Wang et al., this was another sports-related concussion study where they recruited all male subjects, 18 of which were concussed and 15 were controls. This study was special because they scanned both the concussed subjects and the controls at 2 time points. So for the concussed athletes, they were scanned at 24 hours and 8 days post-injury, whereas the controls were scanned at immediately after they were identified as controls and then at 7 to 8 days after the first scan. So they were able to see how blood flow changes are different between these 2 groups. So at 24 hours post-injury, they found decreased blood flow in the superior frontal areas of the brain. And then strikingly, after 8 days, there was diffuse decrease in blood flow throughout the entire brain. And then this image is showing areas where the two groups differ in the amount of blood flow changes they had between the two time points. So moving on to the application of CDR in concussion research, this paper by Much looked at 12 concussion subjects compared to 5 controls. They used this advanced Respirect system, so this is one of those fancy computer-controlled end-tidal CO2 targeting systems that I mentioned about earlier, and they compared normal capnia to hypocapnia and hypercapnia conditions using this system. So they did a twofold analysis. In the first part of their analysis, they just compared the number of voxels with significant CDR in the concussed group versus the control group. And then in their second level analysis, they generated a CDR atlas from all the controls, so a mean CDR map from all of their controls. And then they compared each of their subjects to this CDR atlas and calculated the number of voxels that had increased CDR or decreased CDR compared to this control atlas. So here's an example of their first level analysis with a control subject on the left and a concussed subject on the right. And you can immediately see the difference between the two. The control subject had uniform CDR throughout all of the gray matter regions, whereas the concussed subject had reduced CDR throughout the brain. And this is a general trend they found in all of their subjects, although this finding did not reach statistical significance. And then this is an example of their second level analysis where they compared either a control subject to the control atlas, control CDR atlas on the left, and on the right is comparing one of their concussed subjects to the control atlas. So whereas the control versus control comparison had no significant voxels, the concussed subject definitely had areas that had abnormal CDR compared to the controls. So the same group did a follow-up analysis or a follow-up study on adolescents, where they now included a baseline CBF measurement with pseudocontinuous ASL as well as the CDR experiment. They didn't find any differences between groups for the baseline CBF data, but they did find that using the number of voxels with abnormal CDR compared to the controls is a pretty accurate way of distinguishing between injured subjects versus the controls. And then finally, this paper by Tana looked at 7 varsity athletes within a week after their injury and compared it to 11 controls. They looked at both resting state, blood flow, as well as CDR in response to 5% CO2. This was a functional connectivity paper, so they focused on different networks, including the default mode network, the frontal parietal control network, and the dorsal attention networks. And in all 3 networks, they found increased CDR in the concussed athletes compared to the controls. This was all statistically significant. They also found decreased blood flow in all of these networks in the concussed athletes compared to the controls. However, the blood flow results was not statistically significant unless they only compared the concussed athletes to the athlete controls, which had a slightly higher blood flow. So as you can see, I presented 3 papers on perfusion and 3 papers on CDR, and they all seem to look at different populations, different time points post-injury, and they seem to report different results. So this just goes to show that the field is still in its infancy, and it looks like CDF and CVR are both dynamically changing during the recovery period post-injury. And so more studies are necessary for us to better understand how the recovery process affects these measures. Okay. Are there any questions on what I've presented so far? Okay. If not, I'm just gonna switch gears and go to the final part of my talk, which is our female athlete concussion study. This is in collaboration with Amy Harold. And before I go into details about the study, I just want to give a special shout out to Kaylin O'Connor and Virginia Gallagher for the RA and grad student working on the study. They worked diligently talking to our athletics department to make sure that we're on top of every single concussion that occurred, and followed up with the athletes and made sure that every step of their visit and data collection went smoothly. So the study would not have happened without them. So thank you both. So why do we want to study female athletes? Well, they're rarely studied. If you look in PubMed, I found only 3 papers that focused on female athletes in concussion research. Many others included both males and females, but none of them accounted for the effects of different hormonal contraception or menstrual cycle phase, even though mounting evidence suggests that these are important factors that need to be accounted for in research involving female subjects. So this paper by Wang showed that menstrual cycle phase at the time of injury significantly affects quality of life and neurologic outcome after the injury. And then several MRI papers have also demonstrated that menstrual cycle phase and hormonal levels can affect MRI measures. So here's an example. This report by Craig et al. Showed that estradiol concentration in the blood is positively correlated with functional activity. And then this other paper by Plitzer et al. Is showing that menstrual cycle phase affects functional connectivity in the brain. And then a recent publication by Virginia Gallagher and our group showed, which is a chart review study on the recovery of college athletes after concussion. She reported that there is a significant gender difference in the length of recovery, and also that the correlation between length of recovery and symptom severity was different between genders. Actually, it was significant only for male athletes and not for females. And she also found that hormonal contraception users have lower symptom severity scores compared to those that are not on hormonal contraception. So all of these papers made us think that we should account for these variability, these sources of variability in a female athlete study. So here's, this table shows just the general demographics of our subjects. We recruited 11 injured athletes, and on average they were scanned 7 days after their injury. And we've listed the types of sports that they played. These were matched to 13 controls that were matched in age, handedness, ethnicity, and hormonal contraceptive method. And we scheduled their MRI scans such that they all happened during the same menstrual cycle phase for both the injured and the matched controls. And just to go into a little bit more detail about how we determined that, we divided our subjects into those that don't use any hormonal contraceptives versus those that do. So for the non-hormonal contraceptive users, obviously they have normal menstrual cycle periods, and we targeted either the follicular or the luteal periods of their menstrual cycle, and confirmed our estimate. So the estimation was based on self-report that goes back 3 months before the scan time. And then on the day of their scan, we confirmed our estimation by taking a blood sample and measuring serum progesterone level. So we had 11 subjects that were non-hormonal contraceptive users, and they were all scanned during their follicular phase. For the hormonal contraceptive users, we just divided them into active hormone use versus inactive hormone use based on either their pill pack or the time their NuvaRing was inserted. And again, just matched the scan time of the controls to when the concussed athletes were scanned. So each athlete had 2 visits. In the first visit, we did neuropsych testing and eye tracking, and in the second visit, which is the MRI visit, we collected a blood sample, and also the MRI protocol included a high-resolution structural scan, 2D pseudo-continuous ASL scan, both CDR. We also collected DTI and resting state, but today I'm just going to focus on the ASL and the CDR data. And then this schematic in the bottom is showing the task that we used for CDR. So we did a breath holding task. We began with 30 seconds of fixation and then 12 seconds of cued breathing, where subjects were instructed to breathe in and breathe out at 2-second intervals. And then at the end of their final breathe out, they were asked to hold their breaths for 15 seconds. And this was repeated 6 times for signal averaging. In terms of data analysis, it's pretty run-of-the-mill. We did motion correction within the scan and then co-registered all of the ASL and CBR images to the T1. And then calculated transformation matrices from the T1 to the template space and applied that to the ASL and CDR images. Importantly, for the group analysis, we normalized our ASL maps by each subject's whole brain blood flow. And then for the CDR, we did a first level analysis to acquire the contrast images for the breath hold, and then did group analysis on these contrast images. So here are our ASL results. These were thresholded at a p-value of 0.001 uncorrected for multiple comparisons. We did not find any global blood flow differences between the groups, but we did find both increases in blood flow and decreases in blood flow in the concussed group compared to the control group. So most of our increases were isolated near the superior areas of the brain, including the paracentral lobule, extending into the pre-post central gyri. And then we also had clusters in the inferior parietal lobe. And then for the decreases, these were mostly concentrated in the cerebellum areas. For our CVR results, we only found increased CVR in the concussed athletes, and these were mainly concentrated in the medial regions, including the anterior cingulate, the precuneus. There's also a cluster in left superior frontal. This graph just shows you the average time, the group average time course for concussed athletes in blue versus control subjects in red. This is the percent fold signal change for the CVR experiment. And you can see timing-wise and amplitude-wise, they're quite similar. This was extracted from whole brain gray matter. So unfortunately, because the data is very preliminary, I don't have any conclusive insights on our findings, except that currently we just have mixed RCBF changes found in the brain for our cohort. Our CVR was only, we only found increased CVR in the concussed athletes compared to the controls, and this was primarily located in medial areas of the brain. So this at least seems to be in good agreement with the Tana paper that I presented a few minutes ago. One thing I wanted to point out is in the current analysis, I used a boxcar model to model the CVR response. But if you remember from the previous slide, the time courses really look more sinusoidal. So perhaps a different model will give us a better fit to the data. And then, of course, the usual caveats of MRI studies, we have a smaller sample size, which is why all of the results so far are not corrected for multiple comparisons. And also, unfortunately, we don't have any equipment to and end-tidal CO2, so we won't be able to normalize our data to that. But one possible way around that would be to normalize the CVR signal changes to breath hold duration for each subject, which is expected to be different. So there's a lot more to do with our data. We collected some other data sets, including the blood hormone levels and depression and post-concussive syndrome scores. We also have a working memory task scores. So we'll be looking at all of these in correlation to our blood flow and CVR findings. And then finally, also, we'd like to look at the rest of our data to get a more multimodal view of our post-injury model. So last but not least, I just want to thank members of the Concussion Neuroimaging Consortium at Northwestern. They've given us a lot of valuable feedback and advice. Nurses at the clinical research unit and staff at the athletics department who were great help during the study, which was funded by the Northwestern Women's Board. And of course, thank you all for your attention. I'd be happy to take any questions.
Oh, sorry, Jenny. Sorry, I had my phone on mute. So I'll ask my question again. So I had a question from your next directions were kind of bringing together the multimodal data, and I mentioned that a lot of folks may have multimodal data or may just be looking to see how they can inform fMRI data. And since you mentioned at the beginning and doing a great job of going through our methods that there was that perfusion disruption contributes to the bold signal, I was wondering if you could talk a little bit about how folks could use perfusion data to inform their fMRI data.
Yeah, so both the perfusion and the CVR data, I think the CVR data is especially relevant for bold task, especially supplementary to bold task data, because areas that are unable to support the additional oxygen requirements during hypercapnia in a CVR experiment will most likely not be able to support similar demands in oxygen and nutrients during a functional task. So if you have both CVR data and both task data, you'll be able to see where the areas of compromised CVR are, and combine that with your bold results to see if those are the same areas that are unable to sustain functional activity, and also see if that correlates with any decreases in their cognitive performance. Yeah.
Thank you. Yeah, I think that really helps, because a lot of folks are probably may be inclined to maybe add a perfusion sequence in order to make better sense of their fMRI data that they're getting, especially in some populations that may have cerebrovascular injury. I had another question about the female athlete study results.
Okay.
I was wondering, so we saw there was an increase in CBF in lateral areas, and then an increase in CVR in medial areas. And I was wondering if that kind of trend is seen in perfusion literature from maybe some different injuries.
Yeah, this is really hard to say, because I literally just got these maps like 2 days ago.
Sure.
So I think at this point I don't have an answer for that, and which is why I'm hoping to get my hands on some of the other behavioral data and see if we could explain some of these findings in relation to that.
Yeah, especially the symptom data, because it seemed like the published literature that you presented saw changes when they kind of broke things up that way or correlated with symptoms.
Yeah.
Awesome. And then I was going to ask another practical question. With doing breath holding tasks for capturing CVR, for others that may be wanting to implement this, in our methods, that breath holding task was repeated 6 times. And I was wondering what kind of is the range to do that in order to get a good signal when you're averaging the blocks together.
I mean, it's actually a pretty strong response. I haven't tested, how many blocks are minimally required to be able to detect the CDR. But I think as long as you have at least a handful of properly held breath hold period, I think you should be able to generate a pretty decent CVR map. I would recommend, so hindsight is always 2020, and so now that I've actually done the study, I would recommend if you have the equipment available to monitor both end-tidal CO2 and also to have a breathing belt on these subjects so you can actually, you have a measure of how well they complied with the task.
Yeah. With that, too, you were saying that normalizing the breath hold length may be helpful. And how would you change kind of the instruction, or is changing the instructions to the participant how you would change that length?
No, so I was kind of referring to, just using the data itself. So if you look at the time courses, you can extract the width of each breath holding period. And so what I was thinking is to just get an average of the 6 breath hold runs for every subject, and then using that as a normalization index for each subject's CVR.
Oh, very cool.
But we'll see if it works.
Yeah, that'll be really, really neat. Okay, great. Well, thank you so much for such a wonderful presentation of both methods and how it can be applied to a rehab application for traumatic brain injury. We really appreciate it.
Thank you. Thank you for inviting me.
All right. With that, we'll sign off.
All right.